Welcome back to the Deep Dive. Today, we are focusing on the body's hidden language, the endocrine system.
Absolutely. It's this incredible chemical choreographer.
Yeah, running everything from growth and metabolism to how we handle stress, just fundamental stuff.
And that's our mission today, really. We want to unpack the core mechanisms of how this endocrine control actually works.
So looking at the hormones themselves, how they act, the control systems.
Exactly. Especially that central command, the hypothalamus and pituitary. And, critically, how we figure out what's going on when things aren't working right, the diagnostic.
Okay, let's dive in. Starting with the hormone itself, we often think it causes something to happen, but you're saying that's not quite the right way to look at it.
That's a key point. Hormones are chemical messengers, specialized organic molecules, yes, but they act as modulators.
Modulate.
They don't perform the reaction. They regulate it, adjust it, integrate the cell's response. Think of it less like an on-off switch and more like a dimmer.
Adjusting the volume knob, maybe?
Yeah, that's a great analogy. And where it gets really interesting is realizing it's not just the classic glands doing this.
Right. I was pictured, the pituitary, thyroid, adrenals, the usual suspects.
But the definition now includes what we call non-classical endocrine tissues.
Which broadens the scope massively.
It completely changes the textbook view. Your heart, for instance, makes atrial natriuretic peptide that acts on the kidney.
Wow. The heart as an endocrine organ.
Or the kidney itself. It senses low oxygen, releases erythropoietin.
Which tells the bone marrow to make more red blood cells.
Precisely. Then you have the liver, the GI tract, lymphocytes, even fat tissue, adipose tissue. They're all secreting hormones.
So the whole body is this interconnected chemical network.
Exactly. Which means the action isn't always long distance, like that erythropoietin example.
Okay, so there's a spectrum of action based on distance.
Right. We usually classify them into 5 types based on where they're made versus where they act. The classic one is endocrine.
That's the long-distance call. Released into the blood, travels far away.
Like erythropoietin going from kidney to bone marrow, then things get more local.
Starting with paracrine.
Yes. Paracrine acts locally, but on cells different from the ones that made the hormone. Think of sex steroids in the ovary influencing neighboring cells.
Okay, and then even more local.
Autocrine. This is where the hormone acts back on the very same cell that produced it.
Like a self-regulation switch.
Exactly. Insulin, for example, can inhibit its own release from the pancreatic beta cells that made it. It's a direct feedback.
And then there's action inside the cell itself.
That's intracrine. Synthesized inside, acts inside, never leaves the cell.
Fascinating. And the last one blurs the lines.
Neuroendocrine. Here, neurons make and release hormones, neurohormones.
Like ADH or vasopressin.
Correct. Or epinephrine from the adrenal medulla, which starts as a neural signal. These travel through the blood to distant targets, linking the nervous and endocrine systems directly.
That framework of action is helpful. Now let's shift gears slightly to their structure and what they're made of. Because that dictates pretty much everything else, doesn't it?
Fundamentally, yes. Their chemical makeup determines how they're made, stored, transported, and how they act. 4 main categories.
Okay, what are they?
You've got amines and amino acid derivatives like norepinephrine, epinephrine, and the thyroid hormones, which come from tyrosine.
Okay.
Then peptides, proteins, and glycoproteins. This is the biggest group, everything from tiny TRH to large growth hormone.
The majority. Got it.
Third, steroids. These are all lipids derived from cholesterol.
Like cortisol, estrogen, testosterone.
Exactly. And finally, fatty acid derivatives, like the eicosanoids, prostaglandins, are a key example here.
And how they're synthesized and stored really splits along those structural lines, particularly proteins versus steroids.
Big difference. Protein hormones are made as larger inactive precursors called prohormones.
Okay, why inactive?
It's partly about proper folding and partly a storage mechanism. They sit in vesicles ready to go. When the signal comes often triggered by feedback, the cell clips them into the active form and releases them.
So they're stored and released on demand. But steroids, being fatty, lipid-soluble,
Yeah.
they can't be stored in vesicles, right?
Correct. They're lipophilic, so they just diffuse right across the cell membrane as soon as they're synthesized.
No waiting around. Made and released immediately.
No storage needed for glucocorticoids, androgens, estrogens. The synthesis is the release, essentially.
Okay, so now these messengers are out there in the body fluids, usually the blood. How do they get around? This brings us to transport and protein binding.
Critical concept. It really depends on solubility. Water-soluble hormones, the peptides, proteins, Catecholamines, they travel freely, unbound.
And that affects how long they last.
Absolutely. They have very short half-lives because they're exposed and quickly broken down. We're talking minutes, sometimes less.
Okay, but the fatty ones, the lipophilic ones, steroids and thyroid hormones.
They need help. They can't dissolve well in blood plasma, so they bind to carrier proteins, mostly made by the liver.
And this binding acts like a protective shield.
Exactly. It protects them from degradation and excretion, and it creates a reservoir. The amount of binding directly correlates with the hormone's half-life.
Can you give an example of that difference?
Sure. Take angiotensin II. It's mostly free, unbound. Its half-life is less than a minute. It's like a quick text message.
Gone almost instantly.
Compare that to thyroxine, thyroid hormone. Over 99% is bound to carrier proteins, its half-life, up to 6 days.
Wow, days versus seconds. That's a huge difference in signal duration.
It is, and this binding has real clinical implications. Think about drugs.
How so?
Well, some drugs, like high-dose aspirin, can compete for those binding sites on the carrier proteins.
So they knock the hormone off the carrier.
Right, which suddenly increases the amount of free, active hormone floating around.
And if someone already has high levels, like in a thyroid storm?
That sudden surge of free, active hormone can be really dangerous. It amplifies the effect rapidly.
So understanding binding is key for drug interactions.
Yeah.
Okay, once the message is delivered, how are they cleared out?
Elimination happens mainly through enzymatic degradation, either in the blood or tissues, followed by excretion by the kidneys or liver. Water-soluble ones are cleared fast. The bound ones, the lipophilic ones, are cleared much more slowly as they gradually detach from their carriers.
Okay, let's move inside the cell.
Yep.
The hormone arrives, but it needs something to hear the message. That's the receptor's job, right?
Precisely. Receptors are usually proteins, and they are highly specific. They recognize only one particular hormone or a very limited group. This specificity is what allows TSH to act only on the thyroid, for instance.
And the cell isn't just passively listening. It can change how well it hears the signal.
Yes, through receptor regulation, it's dynamic.
Like turning the volume up or down.
Kind of. If hormone levels are low for a prolonged period, cells often respond with upregulation. They increase the number of receptors on their surface,
Making themselves more sensitive to the scarce signal.
Exactly. It's an adaptive response. Conversely, if there's a sustained excess of hormone,
The cell gets overwhelmed.
right, so it initiates downregulation, it pulls receptors off the surface or makes them less responsive, decreasing its sensitivity to protect itself from overstimulation.
Okay, now the actual mechanism of action. How does binding translate into a cellular change? Again, it depends on the hormone structure.
Entirely. Water-soluble hormones, peptides, catecholamines, can't cross the cell membrane because they're charged.
So they have to knock on the door.
They bind to receptors on the cell surface. This hormone is the first messenger.
And that binding triggers something inside.
Yes. It activates an intracellular signaling pathway, often involving things like G proteins. This generates a second messenger inside the cell.
Like cyclic AMP.
Cyclic AMP, CMP, is a classic second messenger. This molecule then carries the message within the cell and triggers the actual response, like telling the cell to break down glycogen or secrete something else.
So first messenger outside, second messenger inside.
Exactly. Think of glucagon binding to a liver cell. That's the first message. The second message inside says release glucose.
But the lipid-soluble ones, the steroids and thyroid hormones, they don't need a doorbell.
Nope. They're fatty. They slip right through the cell membrane.
So they go straight inside.
They pass through the membrane and bind to receptors inside the cell, either in the cytoplasm or directly in the nucleus.
And what happens then?
The hormone receptor complex then travels to the nucleus, if it wasn't already there, and binds directly to specific DNA sequences called hormone response elements.
So it's interacting directly with the genes.
Yes, it directly modulates gene transcription, turning genes on or off, essentially. This changes the rate of mRNA synthesis, which ultimately alters the types or amounts of proteins the cell produces.
Which explains why their effects can be slower, but also more profound and long-lasting. They're literally changing the cell's protein-making instructions.
Precisely. A catecholamine response via a second messenger can happen in seconds to minutes. A steroid or thyroid hormone response, involving changes in gene expression, might take hours or even days to fully manifest.
Okay, so we have these complex actions. How is the overall level controlled? It's not just constant secretion, is it?
Definitely not. Hormone levels fluctuate constantly. Some follow diurnal rhythms, daily cycles, think growth hormone or ACTH, which peak during sleep.
Right. And others have longer cycles.
Yes, like the cyclic patterns of female sex hormones over the course of the menstrual cycle.
But the master controller, orchestrating a lot of this is the hypothalamic pituitary axis.
That's the command center. The hypothalamus acts as this incredible integrator. It takes input from the nervous system, think pain, emotion, temperature, and even the immune system, like inflammatory cytokines.
And translates that into endocrine signals.
Yes, often by influencing the pituitary gland or hypophysis, which sits right below it. They work as a functional unit.
The master gland, as it's often called. But the connection is different for the front and back parts, isn't it?
Very different. The anterior pituitary or adenohypophysis isn't directly connected by nerves. It's linked via a special blood vessel network, the hypophyseal portal system.
Portal system.
Yeah, it's like a private circulatory loop. The hypothalamus releases tiny amounts of releasing hormones, like TRH or GnRH, or inhibiting hormones, into this portal blood.
And those travel directly to the anterior pituitary.
Right there, telling it to either release its hormones, like TSH or LH, or to hold back. It's all controlled by these chemical signals from the hypothalamus via the blood.
Okay, but the posterior pituitary, the neural hypophysis, that's different.
Totally different connection. It's essentially an extension of the hypothalamus. Nerve axons stretch down from neurons in the hypothalamus directly into the posterior pituitary.
So the hormones are made upstairs.
Exactly. Hormones like ADH, vasopressin, and oxytocin are synthesized in the hypothalamus, travel down these axons, and are stored in the nerve terminals within the posterior pituitary.
And released directly into the main bloodstream from there.
Yes, when the hypothalamic neurons fire. It's direct neural control of hormone release.
Fascinating distinction. Now, overseeing all this release is the concept of feedback, usually negative feedback.
Overwhelmingly, negative feedback is the cornerstone of endocrine regulation. Think of it like your home thermostat.
Okay, so
Sensors in the system detect when a hormone level rises above its set point. This triggers a response to decreased secretion, bringing the level back down.
high levels shut off the production signal.
Precisely. For example, when thyroid hormone levels in the blood are high enough, they signal back to the pituitary to stop releasing TSH and even back to the hypothalamus to stop releasing TRH.
And this can be a multi-layered loop, right? Hypothalamus to pituitary to target gland.
Yes, the hypothalamic pituitary target cell axis is a classic example. The hypothalamus releases RH, the pituitary releases a tropic hormone, the target gland releases the final hormone.
And that final hormone feeds back to inhibit the top two levels.
Correct. Cortisol from the adrenal gland, for instance, inhibits both ACTH release from the pituitary and CRH release from the hypothalamus.
And this has huge clinical relevance, like with taking steroid medications.
Absolutely. If you give someone exogenous corticosteroids, like prednisone, their body detects these high steroid levels. Negative feedback kicks in hard.
Shutting down the natural production.
It suppresses the pituitary's release of ACTH, which means the adrenal glands aren't stimulated and they stop making their own cortisol. They can even atrophy over time.
Which is why you can't just stop steroids suddenly.
Yeah.
The body's own system needs time to wake back up.
Exactly. You have to taper off slowly.
Okay, so that's negative feedback. What about the opposite, positive feedback? You said it's rare.
It is rare because it drives the system further away from stability. Rising levels trigger more secretion, not less.
Until some endpoint is reached.
Right. It's usually involved in processes that need to reach a climax. The classic example is during the menstrual cycle. Rising estrogen levels mid-cycle reach a threshold.
And instead of inhibiting, they stimulate a surge of LH.
Exactly. That LH surge is triggered by positive feedback from estrogen, and it's what causes ovulation. Once ovulation occurs, the endpoint is reached and the system resets.
Okay, so we understand the mechanisms, the control loops. How do we actually assess if this system is working properly in a patient?
Well, the approach can be direct or indirect. We can directly measure the hormone levels themselves, usually in blood or sometimes urine.
Or indirectly.
Or we measure the physiological consequences of hormone action. For instance, blood glucose levels give us an indirect measure of insulin function.
Like the case study you mentioned, Emily, with the blood glucose of 650. Normal is, what, 70 to 110?
Right. That massively high glucose screams insulin deficiency or severe insulin resistance even before measuring insulin itself. Her A1C of 10% also tells us this has been going on for a while.
So what are the lab tests we use for direct measurement?
Blood tests are common. Historically, things like radioimmunoassay, RIA, were used, but they had limitations with radioactivity and specificity.
We have better methods now.
Much better. We now mostly use non-radioactive methods like ELISA, enzyme-linked immunosorbent assay, or chemiluminescence assays. They often use two antibodies to sandwich the hormone, making them incredibly specific and sensitive.
Okay. And what about autoimmunity?
We can also test for autoantibodies. Finding antibodies against thyroid peroxidase, anti-TPO, for example, points towards Hashimoto's thyroiditis, an autoimmune cause of hypothyroidism.
And urine tests, are they still useful?
Yes, particularly 24-hour urine collections. Measuring hormone levels over a full day smooths out the fluctuations we see in blood.
Like for cortisol?
Exactly. A 24-hour urinary-free cortisol level is a key screening test for Cushing syndrome, which involves cortisol excess.
Beyond just measuring levels, sometimes you need to test the response of the system, right? The functional tests.
Correct. These are crucial for understanding the feedback loops. If we suspect hypofunction the gland isn't working enough, we use stimulation tests.
You poke the system to see if it reacts.
Essentially, yes. For example, we might give synthetic TRH and then measure the TSH response from the pituitary. If TSH doesn't rise appropriately, it suggests a problem at the pituitary level.
And if you suspect hyperfunction, the gland is overactive, maybe due to a tumor.
Then we use suppression tests. The idea is to see if the gland's secretion can be shut down by normal feedback signals.
So you give something that should normally inhibit it.
Right. For instance, in suspected acromegaly, excess growth hormone, we give a large glucose load. Normally, glucose suppresses GH release.
But if there's a GH-secreting tumor,
The GH levels will fail to suppress. They keep churning out GH autonomously, ignoring the glucose signal. That confirms hyperfunction.
okay. And beyond blood and urine, we have even more advanced diagnostics now, genetics.
Absolutely. Genetic testing is becoming more and more important. Identifying specific gene mutations can diagnose certain inherited endocrine disorders even before symptoms appear.
Like the RET protein gene.
Yes, finding a mutation in RET on chromosome 10 can diagnose multiple endocrine neoplasia type 2, MN2. This allows for potentially life-saving prophylactic surgery, like removing the thyroid to prevent medullary thyroid cancer.
And finally, imaging, seeing the glands themselves.
Imaging is key. We have non-isotopic methods like MRI, which gives great structural detail without radiation. CT scans are faster, also structural, but involve radiation and often contrast dye. Ultrasound is great for superficial glands like the thyroid, real-time imaging. And DEXA scans measure bone density, often affected by endocrine issues like osteoporosis.
Then there are the scans that show function, not just structure.
Right, the isotopic methods or nuclear medicine, these involve giving a tiny amount of a radioactive tracer that's specifically taken up by the gland or tissue of interest.
Like radioactive iodine for the thyroid.
That's the classic example. The scan shows how actively the thyroid tissue is taking up iodine, revealing nodules or overall gland function.
And PTCT, that combines both.
Yes, KTCT is incredibly powerful. It overlays the functional metabolic data from a PET scan, using tracers like radioactive glucose, onto the detailed anatomical map from a CT scan.
Providing both structure and function in one image. Useful for cancer.
Very useful, especially for finding metastatic disease like thyroid cancer that has spread and might show up as metabolically active spots on the PET scan.
Okay, quite a toolkit. So let's try and wrap this up. What are the core takeaways from this deep dive into endocrine control?
I think the main points are these. First, the endocrine system uses these specialized chemical messengers, hormones, as modulators, not direct actors. Their structure, amine, peptide, steroid, fatty acid dictates their synthesis, transport, and mechanism of action.
Right. Water-soluble versus lipid-soluble makes a huge difference.
Huge. Second, control is highly centralized via the hypothalamic pituitary axis, which integrates all sorts of body signals. And this control predominantly relies on elegant negative feedback loops to maintain balance.
Like that thermostat keeping things stable.
Exactly. And third, diagnosing problems requires a sophisticated approach combining direct hormone measurements, dynamic function tests, stimulation and suppression, and increasingly, genetic analysis and advanced imaging like PTCT.
It really highlights the complexity from, a single amino acid being modified into a hormone to these intricate feedback loops involving the brain, pituitary, and target glands. It's a remarkably fine-tuned machine.
It really is.
Yeah.
And slight disruptions anywhere in that chain can have significant downstream effects.
So a final thought for you, our listener, to ponder. We touched on how the hypothalamus integrates signals like stress, pain, and immune factors like cytokines. We also know that chronic low hormone levels can lead to upregulation, making cells more sensitive. Now, consider the opposite. Chronic stress, that's a constant barrage of neuronal and cytokine input to the hypothalamus. How might that sustained input eventually trigger downregulation in a key pathway, maybe like the HPA axis involved in the stress response itself?
That's a great question.
And if cells become less sensitive due to that downregulation, what might be the clinical consequences of the body essentially becoming numb to its own stress signals over time? Something to think about.
Definitely food for thought, connecting those basic mechanisms to broader physiological states.
Thank you for joining us for this deep dive into the mechanisms of endocrine control. Go forth and apply this knowledge.